Hello everyone, welcome back to Hang Education. So today in this video, I will be continue explaining about the genetics and inheritance. So this would be my part two video. So before continuing, I would like to recall from my previous video. So from the previous video, part one, we cover about the key definition, including dominance, recessive, genotype, phenotype, homozygous, and heterozygous. And then after that, we slowly proceed towards the working examples about to identify which are dominant, recessive, homo, hetero, geno, and pheno. And after that, we go on to genetic diagram. So this would be a very systematic way of doing or like calculating for the Punnett square, calculating genetics using the Punnett square. And after that, we go over the pedigree, which is a diagram show a family tree for an inheritant characteristic. And we learn how to sort for the pedigree diagram, as well as we also talk about test cross. Test cross basically is to identify a genotype of a character where it is expressed by a dominant phenotype, right? So by crossing with a homozygous recessive for the same trait and to be able to identify whether this character is homozygous or heterozygous. So this would be a test cross. And last but not least, we talk about a codominance where both alleles are expressed in the phenotype, and we stop at a flower colors in Snapdragon. So today I would like to continue with a blood groups, or let's just say blood typing, or like blood groups calculation. So as we all know that, and since most of us already familiar, there are four different blood groups. So let's say blood group A, blood group B, blood group AB, and blood group O. This would be the, let's say, phenotype. But when we do about the blood groups, there are three different alleles, which are IA, IB, and IO. But in some textbook, this is also expressed as small letter I. So now let me write a sentence which talk about this blood groups, codominance character in blood groups. So which is the alleles IA and IB are dominant over IO, but they are codominant to each other. So meaning that the alleles of IA and IB, they are dominant over IO, but then they are codominant to each other. So what does it mean is that for blood group A, there can be two different alleles, or let's say two different combinations. So for blood group A, it can be either IA, or it can also be IA, IO. And for B, similarly, it can be I'm B, or it can also be I'm B, IO, right? But for AB, it will be IA, I'm B, and for O blood, it's me IO, or let's just say small letter like I, right? So this would be the genotype and phenotype of the blood group. And let's just say, let me go straight directly to like where we can get a blood group O from a mother, blood group A, and blood group B. So it is a very simple way, right? So this would be the example calculation. So let's say we will cross with parents A and parents B, and then the offspring getting O offspring, right? So let's say we got a blood group A. Okay, so we have heterozygous blood group A and blood group B. Okay, here we have blood group A and blood group B, and their genotype would be IA, IO, and here IB, IO. So same thing, similarly, we will have two different things. So let's just draw a pedigrees. So now we got a pedigrees, and then we have IA, IO, and IB, IO. So the first one would be IA, IB, here IB, IO, and we have IAIO, and here IAIO. So from this example, we can see that let's say mother of blood group A and father with blood group B can produce a offspring with blood group O, since both of them can be heterozygous and they may give birth to a O blood baby, or let's say O blood children. So here we have IAIO is to IBIO is to IAIB is to IOIO. So it will be 1:1:1:1. So this is like a way how we calculate for a blood group as well. So let me go over one more time about blood group, which is a part of codominance as well. So IA and IB are dominant over IO, but they are codominant to each other, and we can see that. So there are four different phenotype, blood group A, B, AB, and O, three different alleles, capital three different, right? IA, IB, and IO. But when we combine into a possibility, then we got like six different pairs. So for blood group A, it can be IAIA or IAIO, and for blood group B, it can be IBIB and IBIO. For blood group AB, it can be IAIB, and for blood group O, it will be IOIO. And then we can see a calculated example where we got a heterozygous blood group A mother and heterozygous blood group B father, and then these are the probability of offspring that they can get, and that is no surprise. Sometimes some of my students, they would ask me like, oh, so yeah, like my parents is A and my mother is A and my father is B, but my brother or my sister is blood group O. Can it be possible, or can that be even make sense? So then we can show with this diagram, and we can show that okay, this makes sense, and then like they can give to all the blood group, like blood group A, B, AB, and O as well. So this will be about codominant. So codominant is when both alleles are expressed in their phenotype. So in this case, it would be blood group AB, right? So it's not either A or it's not B, but it's AB. So it means both the characters are expressed in their phenotype. Okay, so moving on to next part, which is about sex determinations and sex-linked diseases, or let's say sex-linked conditions. So starting off with sex determination. Sex determination is very simple, very easy. It's to determine a man and a woman when they have sex or like have sexual intercourse and get pregnancy, and then like what is the probability of that their child will be a male or a female, or let's say a boy or a girl. So now this is a kind of calculation. So for mother or for male, let's say mother or female, so they are represented by XX chromosome, and for father or male, we represent them by XY. So this is also the another easy calculation. So we got mother XX and father XY, and again we will have the Punnett square to calculate for that. So again, similar, same thing. So you draw a Punnett square. We got x and xy. And here we can see x, xy, xy. So it's a 50% chance, right? 50% x is to 50% xy. So meaning that 50%, so a couple or every single pregnancy have a chance of giving birth to a boy is 50%. Similarly, for a female or a girl, it will be 50%. So it's a 50-50 chance of giving birth. But it doesn't mean that if a couple have four children and two of them are male and two of them are female, it doesn't mean like that. So each pregnancy or like each birth, it is 50-50 chance of getting a boy or a girl. So this would be a sex determination, right? So let's me review one more time. Female, they are represented by XX or XX chromosomes, and for male, it will be XY. So Y is only from the sperm, or it's Y is only from the male, and X chromosomes are provided. It can be provided from both male and female. So this is like how we can determine, and that's how it's come up to the point that Y chromosome or father can determine whether their child will be female or a male, because you can imagine, right? from a mother or from a female, both all of our egg cells will have all of our egg cells, all of our egg cells will have X chromosome, but for the father or for male, half of his sperm will be containing X chromosome and half of his sperms will be Y chromosome. So if the fertilization happens between an egg cell with an X chromosome and an X chromosome sperm cell, then they will produce a girl. But if a father give a sperm containing Y chromosome, then they will produce a boy or they will give birth to a male baby, right? So this will be a sex determinations. And after that, we have to look about sex-linked condition. So basically, sex-linked condition or sex-linked diseases, they are carried on the X chromosome. That's how its name begins. So we get sex-linked diseases, or let's say sex-linked condition. So it's mean the alleles or the gene is carried on the sex chromosome. The gene is carried on the sex chromosomes, right? So it can be either X or Y chromosome, and we can divide it into two, right? So first would be Y link conditions. So what does it mean by Y-link condition is that the chromosome is carried on the X chromosome, right? So the alleles or the genes is carried on the X chromosome. So let's talk about hemophilia, right? Hemophilia, this is a very typical example. So hemophilia is caused by recessive allele. So for a part of female, so let's say for female, there will be H capital H, X capital H, H capital H, H small h, and H capital H, oh, sorry, H small h and H small h. So this would be a three different possibility that a female can have. So the first one would be a normal female. Second one is also a normal female, but we can also call her as carrier because she has one allele that can lead to a condition or that can lead to disease. And the last one is affected female. So meaning that she will have the hemophilic condition. So let's look at for the male. So for the male would be H capital HY and H small hy. So this would be a normal male. And the second one is affected male. So what you can see is that if the condition is a sex-linked chromosome or sex-linked diseases, for a female, there is only a 1.3 chance of females having the condition, but for a male, it will be 50-50 chance of having a condition. So in this case, we normally see that in a population, if a lot of male get affected by that certain condition, it can be a sex-linked condition or like sex-linked diseases, sex-linked characteristic, right? Because most of the male gets affected because like in this case, the Y chromosome doesn't carry any condition, any genes, but X chromosome is carrying and H chromosome is from the mother, right? So let's show some example. Why do we call normal female a correct carrier for a heterozygous, right? So we have a, let's say here, I will draw here. So we have one carrier female, so it will be H capital H and H small h. And then let's say normal father. So when we look at it, both mother and father is fine. They are normal male and normal female. They do not have, they do not have any condition. But let's see what's happened. So normal female, again, normal female, but she is a carrier. Here, normal male, but here we got affected male. So you can see that that's why we call a heterozygous condition in female a carrier, because she has the potential, or she has the genes that can lead to one of her son to become a, to have the condition, right? Because she is carrying the one genes, or like carrying that alleles that can pass on to her offspring. So that's why it is a uh carrier female, right? Carrier female. So overall, this will be X-linked condition, where the condition, or where the genes is carried on the X chromosome. And you can see in this case, for a female, they have a chance of, let's say, 1.3% chance of 1/3 chance of getting a condition, but for a male, they would be 1/2, which is 50%. So now let's look at another case, which is wine-link disease or wine-link condition. So here, I will just use the same example with capital H and small h is a affected person. So wine-link disease or wine-link chromosome. So for female, since they don't have Y chromosome, they will only have XX, and all of them will be normal female. So it's mean for a female, they got a 0% chance. But for a male, so we can see that again, this will be like this and like this. So the first man is normal male, but for a second one, it is affected male. So we can see that in both condition, either if it is X-linked diseases or Y-linked diseases, male, they are 50-50 chance. But for female, if it is a Y-linked condition, they have 0% chance of getting the condition. But for a X-link, they have 1/3 chance of getting the condition. So this will be about a sex-linked condition. So I'll repeat one more time. So for the sex determination, it will be to determine whether the offspring produced will be a male or a female, or let's just say a boy or a girl. And for a sex-linked condition or sex-linked diseases, it's all about that genes or the allele is carried on the sex chromosome, and it can be divided into two, either X-linked condition or Y-linked condition, right? If it is X-linked condition, again, most male will be affected, and some female can also affect it. But if it is a Y-linked conditions, or if it is Y-linked diseases, none of the female will be affected, and only male will get affected. So in general, we can say that when you look at a population, or when you look at a pedigree in some family, and if you see that a lot of men are having the condition, then we can assume that, okay, this could be a sex-linked condition, or that could be a sex-linked characteristic. And then we can also, like, do a further calculation through a Punnett square to make sure that. So this would be overall about the inheritance and genetic that I would like to share. And I think this will also be the main important concept that most of you, every one of you who are taking IGCSE biology would be needed for your exam. So I think this video helps with your genetic calculation. And let me go over one more time. So for both my part 1 and part 2 video, so it's all covered about 6 key definition, which I think would be the very essential definition that you need to be familiar with to be able to solve the genetic problems with a full score. And then after that, we start off with a simple monohybrid calculation. So monohybrid means when we are looking at the one character only. And after that, we look at it, we look about pedigree, so which is a family tree, and we can identify whether a man is affected or a female is affected. And then we can look at their offspring, and also from the offspring, we can look at their parents. And also we talk about test cross. Test cross is to identify whether a phenotype of a condition is homozygous or heterozygous by crossing it with their homozygous recessive traits. And then we move on to codominant. So codominant is when both alleles are expressed in their phenotype. And last but not least, we talk about the sex determination and the sex-linked condition. So this will be all about for this video. And hope this helped you a lot. And also, if you have any questions, and one more thing I forgot to mention, if you need any worksheet or any other exercises regarding to this topic, you can send email provided, or either you can talk to me through my Instagram account. So, but I prefer email, would be very professional too. So hope you see you again. Hope to see you all in next upcoming video, and hope this video helped with your studies too. And have a great day, and thank you so much. Bye bye.